research that's being done on that. So I'll do my best to kind of make it accessible. Don't feel bad if you feel like you're struggling a little bit to understand it, because I've been struggling to understand it. But it's really exciting stuff. So let's see here how we're doing. Okay. So I'm going to go ahead and introduce myself. I'm Dr. Ron Hunninghake. I'm the Chief Medical Officer here at the Riordan Clinic. Been here 29 years. And we're doing part 2 of a 3-part lecture series on intravenous vitamin C. Our first part related the research that the Riordan Clinic has done over the last 25 years on IV vitamin C, and specifically how it relates to the adjunctive care of cancer patients. This is going to be broader in the sense that we're looking at the effects of the growth of chronic illness in our society, in our culture, and how one of the common symptoms of just about all chronic illnesses is fatigue. And so we thought college, I never really thought I'd be using that much chemistry. Doctors learn biochemistry in medical school, but the actual practice of medicine doesn't involve that much biochemistry, surprisingly. Research does. But here at the Riordan Clinic, with the emphasis on intravenous vitamin C and on finding the root causes of chronic illness, we have, shall we say, dived deep into the biochemistry of vitamin C. And so we'll start out today with this first section. Vitamin C is nature's redox buffer molecule. So Dr. Riordan was himself big into the whole concept of energy. As a matter of fact, in the early days of the Riordan Clinic, which used to be the Center for the Improvement of Human Functioning International, he came up with a very broad definition of health as being the reserve to do what you want to do and need to do with energy and enthusiasm. So if you're feeling more energy and more enthusiasm in your life, you're probably moving in the direction of better health. If you're not, then hopefully today I'll give you some ideas about what you can do to help improve your energy output. And vitamin C is always a good idea, and you'll see why as we get more into this. And so here's the vitamin C molecule. And I know I'm going to be showing you a lot of molecules, and no one's ever really seen a molecule, but we infer through science what the shape of the molecule is, what it's composed of, the elements and whatnot. But vitamin C is such a fundamental thing, and people kind of intuitively know it's very important, but today you're going to find out why it's so important in terms of our cellular health and the organelles that make up our cells. So it was a tough molecule to discover. Dr. Albert St. Gorgie, who got the Nobel Prize for his research on the discovery of vitamin C, for a long time called it hexauronic acid. He knew, what he studied was the adrenal glands. And so it turns out that the adrenals and the pituitary are the 2 highest sources of vitamin C content in the body. So that should tell you something about where is vitamin C important and what does it do? You know, of course, the adrenal glands, the pituitary, they're all about hormones, and hormones are about helping us adapt to stress and to coordinate what's going on in the body. And so you'll begin to see how vitamin C is such a crucial part of this story. So that's really the first major concept. What makes vitamin C so special? It's a redox buffer molecule, and we'll see exactly what that means. The beginning of wisdom is to give things the right name. And so I'm going to start out the lecture by talking to you about vitamin C, which is really known as ascorbic acid, and it is an acid. And if you write a little formula, it's ASCH2 down at the bottom, and you can see why, because there's two hydrogens. These are what's called protons that are available on the basic ascorbic acid molecule. And maybe you remember from chemistry that an acid donates the protons to the solution. Whatever you're putting the acid into, it donates its protons to make the solution acidic. So basically, vitamin C is, when you put it into water, a proton donor. It works in another way, and I'm going to come back to this idea of buffering. You know, in pH, you can buffer a solution with a weak acid so that no matter what you do, it will restore it back to neutral. So for those of you that have stomach upset and you take an antacid, that's a buffer to help you. If you've got too much acid, it will bring your pH up. If you're too alkaline, it'll bring it down and more towards the middle. And so we're going to be talking about vitamin C as a redox buffer. And it's a molecule that is acting as a redox buffer, and we'll get to exactly what that is. So a little bit more about the names. When you put vitamin C, ascorbic acid, into water, it becomes the ascorbate ion. It dissociates into the vitamin C molecule plus a hydrogen in the water. And so this is where one of the hydrogens come off, and you can see up here how it's the ASCH minus, it's minus one hydrogen, is you can actually mark that on the molecule. And that pK constant is a dissociation constant. It tells you how readily this particular acid dissolves into water. So vitamin C absorbs very quickly into water. It has a fairly low dissociation constant. So the lower the number, the greater the tendency of the hydrogen to dissolve into the water, making the water more acidic. Now, there is one other form of vitamin C in water, and that's called the ascorbate dianion, and that's where both hydrogens come off. But notice up there that the pK of that is like 11.8 or almost 12, so it's a very high pK. So this doesn't happen very much. When you put vitamin C in water, it's going to be in the form where just one of the hydrogens has come off. So then, when you're looking at these various molecular symbols, you can see the little E over here. This is an electron. So when vitamin C, when it's dissolved in water, when it donates an electron, that means it creates a vitamin C free radical. So this is what the dots are. The dots are showing you that the vitamin C now, once it's donated the electron, it's already donated one of the protons. If it donates one of its two electrons that it can donate, it becomes a radical. So now we're starting to talk about redox. So there's a whole new field of medicine. It's not a new field, it's a very old field, but redox medicine is where we're looking at how these redox reactions are very important in terms of our health. So there's a little mnemonic that chemistry students learn called oil rig. And oxidation is loss, that's oil. Rig is reduction is gain. So whenever you have a loss of an electron, some other molecule has to gain it. So that's why redox always happens in two halves. An electron is lost, and then that same electron is gained by some other molecule. So if it's an oxidizing agent, it promotes electron loss. And if it's a reducing agent, it promotes electron gain. So antioxidants promote electron gain. So some people say that vitamin C is one of the most important antioxidants in the body. So if we go back to this, you can see where this form of vitamin C, the ascorbate ion, is very willing to donate its first electron to some sort of radical, free radical, and that will neutralize the free radical. So this is actually showing you that the dissociation constant, which of this radical in terms of losing the other hydrogen, is very, very low. In other words, this doesn't stay in that radical form very long. So the particular molecule down below here, this ASC with the dot and the minus sign, that is the ascorable radical. That's the way you mostly find vitamin C. So probably many of you have never even heard this before. It may not even matter when you're taking vitamin C. But from a theoretical perspective, it does make a big difference as we get further into the discussion of how does vitamin C relate to mitochondrial energy or my energy, because your energy is dependent upon your mitochondria. Your mitochondria constitute 1/10 of your body's weight. Almost all of your cells have many, many mitochondria, like the brain cells have as many as 5,000 to 10,000 mitochondria per cell. Your heart cell, which is beating 80,000 times a day, that's got 5,000 mitochondria per cell, if you're healthy. When you're not healthy, you start to lose mitochondria, and that's where you're losing your energy source. So don't get too discouraged. We're not going to have you memorize. There's no test at the end of this, but this tells you that there's a whole family of ascorbates. And so vitamin C ain't just vitamin C. You have to look at what kind of a solution it's in and what's happening, what are the free radicals it's trying to deal with. But in general, the ones that we're going to focus on the most is this little guy down here, the ascorable radical. This is the way vitamin C does its electron buffering. Electron buffering, or redox buffering is another way of putting it. I'm going to go into that. So no need to remember all these different ways that vitamin C does exist, but there is a family of ascorbates, and oftentimes you'll hear the word ascorbate. And so it could be any one of these, and you have to be specific. But the ascorbate radical, or the ascorable radical, that's the one that is most active in the body. This is the one that interacts in our cells and in our mitochondria to kind of help keep them functioning properly. So after dissolving into water or blood, ascorbate is free to lose or donate an electron, which makes it very unique, to be able to either give an electron away or to take an electron back. That gives this molecule incredible flexibility and utility within the body. And that's why when I get a little further along, I'm going to say this is one of the most important molecules for maintaining health. And it's so ironic that millions of years ago, there was a mutation in our genes such that we can no longer make vitamin C. We have to take it either as a supplement or within our food. And so eating healthy food is a great way to get vitamin C, but if you're under stress or if you're sick or if you have oxidative stress of any kind, extra vitamin C makes a lot of sense, and we're going to find out why. So these are the two main forms that you might want to think about. The ascorbic acid, if you're taking a pill or you're putting a ascorbic acid powder into water, this is the form that it's in. And then it dissolves into the water, and very quickly it can shift into this ascorable radical anion. It's a negative anion. When you ionize, there's a positive and a negative side to it. And so this is a negative anion. But we just sort it and just call the ascorable radical is the best way to think of it. There's even another name, because the very final way that vitamin C, the way it's excreted by the kidneys, is this dehydroascorbic acid, which we're going to talk about more in just a minute. It's very important in terms of the cancer research. But once it gets to that stage, the kidneys very quickly excrete it. So this is the problem, is that when you take vitamin C as an oral supplement, the powder or the pill, its half-life is about 30 minutes. So after about 3 to 4 hours, any vitamin C that you've taken in this form, it's pretty much out. It goes out fairly quickly, and that's why frequent dosing of oral vitamin C is a good idea if you're wanting to get the benefits of vitamin C. Just taking a little 500 milligram once a day may not be enough if you're dealing with a chronic illness. The main factor that I'm emphasizing in this lecture is that this ascorbyl radical is very important, partly because it's not overly reactive. It will hang around. It will not immediately, like most free radicals, they are kind of, they're stealing electrons from other molecules, and they can be dangerous. Vitamin C is a very domesticated or very kind free radical. And it can go either way. It can donate or accept an electron. So that makes it very, very utility-wise, very important to the body. It's like a Swiss Army knife molecule for the body. And one of the reasons for this is, here's the ascorbyl radical, and that electron, that extra electron that it's got, kind of migrates through 6 different atoms. And it's this. I tried to show that with the little dotted lines. And the chemists call this a pi system. And this is part of what makes the ascorbyl radical relatively unreactive. It's sitting there waiting to do what it needs to do, but it's not going to jump in prematurely and get shifted to something else. So now we're really talking about redox. So redox is 2 half reactions, reduction and oxidation, or oxidation and reduction. And so this is where one half either gains or loses a proton, and the other half either gains or loses an electron. So that's redox, and it's happening all the time. And you can see a little redox reaction here. If you have 2 hydrogens and it reacts with oxygen, here's the 2 hydrogens, and here's the 2 oxygens with their little electrons. Those dots are the electrons. That forms 2 waters. So now you're seeing a little bit of chemistry in action. But what actually makes that happen? Well, one of the 2 sides is more what's called electronegative. And electronegative means that it's more attractive. It wants the electrons. It's got a very high electronegative potential. So oxygen, which is the second part of my lecture today, which we're going to talk a lot about oxygen, is the second most electronegative molecule in nature. That's what makes oxygen so very important in terms of redox and in terms of your mitochondria. Oxygen, as you know, is basically what you need to have to create a fire. And basically a fire is oxidation in action. So our mitochondria are like little fires or little furnaces, little energy generating engines in our cells that are literally playing with fire. And so this is the significant thing that we're discovering about mitochondria. You've got to keep the fire controlled. You've got to keep it in balance. And so this is where balancing these different, very powerful redox reactions can give you a lot of energy, or it can spell disease and doom and cancer and a number of problems. So here's the periodic chart. It's showing you that electrons in a redox reaction, they're always moving in the electronegative direction. And up there, oxygen is the second most electronegative molecule on the periodic chart next to fluoride. So it's what is the motive force that drives the reactions that occur in your mitochondria. Now, this guy is a very important name in the field of vitamin C thinking. Erwin Stone wrote a book. He wrote many different things, but the book was called The Healing Factor. It's available online free at the Vitamin C Foundation website. It's a very wonderful little book describing how vitamin C was instrumental in the evolution of life on this planet, on the Earth. And so he says here, on a molecular basis, the whole living process is nothing more than an orderly flow and transfer of electrons. So at the submolecular level. Therefore, having an abundance of a substance like ascorbic acid present in living matter makes this orderly flow and transfer of electrons proceed with greater ease and facility. Vitamin C acts substantially like an oil for the machinery of life, and this was discovered by nature billions of years ago. I'll come back to this quote later, and you'll kind of see how it goes. And so again, here's that ascorbyl radical. Very unreactive. It's very domesticated. It is so unreactive that it does not react with oxygen in any way. That's kind of true, but it's kind of not true. And I'm going to, this is the part that has been an amazing discovery to me. How does vitamin C have anything to do with oxygen? And yet it's a very important cofactor in the orderly, shall we say, burning of fuel. Our body, you're eating food today, your body has to burn that fuel, not as a fire, but as a biochemical fire. And what you want is something that keeps the fire going in a way that it doesn't do damage to the rest of the cell. So this is why we refer to vitamin C as a buffer. A buffer solution is a solution which resists a change in the pH when small amounts of acid or base is added to it. So at the mitochondrial level, and I'm going to show you some of the anatomy of a mitochondria, but what vitamin C is doing is it's keeping the flow of electrons orderly. And if it starts to get a little bit out of hand one side, the vitamin C will pull it back in. It buffers the electron flow back into more of an orderly flow. If it gets too excessive to where it could do some oxidative damage, vitamin C brings it back into balance. So this is a redox buffer molecule. And so the way this is a little definition, a redox molecule such as a Scorable Radical, when added to an unstable mitochondrial matrix, donates either a stable source of available electrons or accepts potentially dangerous or damaging electrons from any species of any excess of reactive species that are originating from uncoupling situations within the affected electron transport chain. Now, I just throwing a whole bunch of new words at you, but we'll get into what an uncoupling situation is, which causes electron leak and aging and damage to the mitochondria. We need the vitamin C to help protect us from it. And so this is why I refer to the Scorable Radical as a bioxidate. It can donate or accept an electron whenever necessary. And so here's just a real quick picture of an electron transport chain. It's present in the membrane of your mitochondria. I'm going to talk more about this as we go along, but I want to just, since I mentioned the word electron transport chain, I wanted to show you it's basically a membrane. You can see the structure of the membrane, the little phospholipids there. And then these enzymes, complex 1, 2, Q, 3, cyto C, and 5, these are all important enzymes that are embedded in the membrane of the mitochondria. And this is where the electron transfer occurs. It's very, very important in terms of energy production. Okay, so basically, this is basically the exchange that goes on in water. The vitamin C can either gain an electron or it can lose an electron. So this is nature's stress adaptation molecule. I've also called it Redox Oil, after what Irwin Stone said about vitamin C being like an oil. Can you imagine what would happen to your car or to your lawnmower if you started it without putting any oil in it? I actually did that once with a new lawnmower. I got about halfway with one starting to mow and it stopped because it kind of froze up. You need to have oil in your engine in order to take away the friction of those moving parts. And that's a very good analogy to how vitamin C works at the cellular level. Now, everyone's kind of got an idea of vitamin C as this buffer molecule. It's very important. Now let's go on to a little bit about, we're talking about mitochondrial energy, how vitamin C relates to that. I'm going to introduce a concept to you that maybe you've never heard of, and that's called activated oxygen. And I'm going to show you that the oxygen that we breathe from atmospheric air is not activated. It is not as reactive as you think it would be. Matter of fact, you can put a lighted match into a hyperbaric chamber, and it will not. There has to be something there for the oxygen to burn. Oxygen by itself, just sitting in a chamber, is not that reactive. It has to be activated. So part of what's going on in the mitochondria is the activation of oxygen in order for it to receive the electrons. Because if there's nothing to receive the electrons, there's nothing to pull the electrons along. And so there's no motive force there to create energy. So this is the title of this second concept. Activated oxygen creates mitochondrial electron flow. So this goes back a mere 4.567 billion years ago to the geological start of the planet. I know there are different ideas about evolution and when things started, but geologically speaking, we're going to talk in that realm, 4.56 billion years ago of what they're estimating. 3.8 billion years ago was when the atmosphere began to form, but it was pure carbon dioxide. And even with just that, life did begin, and there were a number of single-celled organisms that were called prokaryotes, and they had a membrane, and they had the ability to make proteins on the inside, and they had a nucleus, DNA nucleus, but they were pretty simple. And that predominated for a long time until photosynthesis as a process came about. And then in photosynthesis, the sunlight is able to split water into hydrogen and oxygen, and that oxygen is released. The hydrogen is coupled to the carbon of carbon dioxide, and you start making sugars. And you make all kinds of molecules. That's carbon chemistry. And so that was the beginning with photosynthesis. But this toxic oxygen, oxygen is really pretty toxic. It built up over millions and millions of years in the atmosphere, and about 2.5 billion years ago, they estimate that we had an oxygen atmosphere, and it was around that time that eukaryotic life emerged. So I've mentioned prokaryotic as being a very simple single-cell organism. Eukaryotes have organelles that use oxygen. And so we're going to come back and talk about those in just a minute. And I want to tie the vitamin C into this whole story. And in Erwin Stone's book, he basically says life couldn't have evolved without vitamin C. These are some of the things that we talked about last time when we were going over the benefits of vitamin C in cancer care. But I want to focus on these two, that vitamin C helps to relieve cellular hypoxia, and it restores aerobic metabolism, and it restores dysfunctional mitochondria, or mitochondrial functioning that's not working properly. It also improves apoptosis, which can be helpful in a cancer situation. So these two concepts, cellular hypoxia and mitochondrial functioning, what does vitamin C have to do with that? Well, it's very interesting. We really shouldn't even call vitamin C a vitamin because it's really not a vitamin, because in just about every other animal except for humans, primates, and guinea pigs, the vitamin C can be synthesized by the body. So is it a hormone? Not really. Is it a lipid? No. It's not a carbohydrate. It's not a food. It's not a sugar. It's not a byproduct. It's not an enzyme or a precursor. Maybe you could call it a cofactor, but not really. Not a cure-all, almost, but not really. So what is it? So we're back to our redox buffer molecule. This, to me, is, as I keep thinking about what is the vitamin C doing, because we give a lot of vitamin C here at the Riordan Clinic, and we see very good results with it, but a lot of people wonder why. Why does it work? And of course, there's new science emerging that you can give high-dose vitamin C to patients with life-threatening sepsis and pull them out of the nosedive. So there's a lot of things that vitamin C can do, and we know it helps in many ways, but how does it work? See, this is what the scientists, the conventional doctors are waiting for. How does vitamin C do what it does? So now, building upon our first part of this talk about the Scorable Radical, we see that vitamin C is still an excellent electron donor. So it's an antioxidant. It may be the premier antioxidant. And so the way it does that, if you look at the little diagram over here, so here's our the vitamin C that's been dissolved into water, and it donates an electron, and now it becomes the Scorable Radical. It donates the electron to a FR, means a free radical, and that gives the free radical 2 electrons, and that's what it wants. It needs 2 electrons in the outer shell, and then it feels satisfied. Otherwise, so some people refer to free radicals as electron stealers. They're promiscuous. They'll steal an electron from, let's say, a cell membrane, and it'll cause oxidation in the cell membrane, and that will disrupt the receptors and cause all kinds of havoc. So if you have an excess of free radicals in your body, that's oxidative stress. And there's all kinds of things, all kinds of toxins, infections, chemicals, radiation, a number of things. Just stress itself, too much adrenaline can cause oxidative stress. And so this is where vitamin C comes to the rescue and donates its electron. So here I have another diagram that's showing that iron is an example. There's a form of iron called Fe3, ferric iron, that when the vitamin C donates the electron to the iron, then it converts it to the ferrous form, Fe2. That's the reduced form of iron. That reduced form of iron can interact with atmospheric oxygen and create something called the superoxide. So you can see here where there are two electrons that are not paired, unpaired in regular O2, and then what the iron will do is donate one of its electrons that it just received from vitamin C and give it to the superoxide. Then the superoxide will interact further in the bloodstream or wherever it's at and create hydrogen peroxide. So hydrogen peroxide, as you know, is a reactive oxidant. If you put it on a wound, it will turn white and bubble. We just recently injected a wart on my forehead, and the vitamin C converted to peroxide, and the wart turned immediately white, and after 2 or 3 minutes it turned black, and 3 days later it flaked off, and now it's healing wonderfully. So we think that this is one of the ways that vitamin C is beneficial in terms of its pro-oxidant actions within the body. This was actually proven at the National Institutes of Health back in 2007, where they gave ascorbate in pharmacologic concentrations. In other words, they gave it IV, or in this case where they were checking, they did it in, I think in rats, they gave it intraperitoneally, but they gave it like 15, 20,000 intraperitoneal. And they were able to demonstrate that hydrogen peroxide was formed in the extracellular space. So this was significant. This is kind of a picture from that National Institute of Health article showing these various steps. And what I'm going to do is kind of break it down in terms of showing you the various reactions and how this hydrogen peroxide is a big deal, because it can go on and form the hydroxyl radical, which is the most powerful soluble free radical in the human body. And so these reactive species, reactive oxygen species, a lot of people are talking about them. Are they our friends or are they our enemies? Are they creating problems? Are they creating disease? Or are they what's helping us to defend ourselves against diseases like cancer, like chronic fatigue syndrome or Lyme disease or any kind of chronic infection? So what's actually happening here is here's our the vitamin C that we've taken this powder. It's in water, or we put it in an IV and we've infused it through the vein. And so this ionized ascorbate, it forms into the radical, and this is the radical here, the ascorable radical, and then that activates oxygen to the superoxide state through iron. These are the Fe2 and the Fa3 that I was talking about. Here's oxygen. It's being activated through an enzyme. Here it's GPX, which is glutathione, and catalase, and then that's actually neutralizing the hydrogen peroxide that gets formed. So when you give IV vitamin C, even though it does transiently create some hydrogen peroxide in the bloodstream, it's gone right away because of these two enzymes, catalase and glutathione. And so this is what they were able to show is happening when they gave high-dose vitamin C. So the question that I had as I was thinking about this, how did that actually happen? Is there enough iron in the blood to make that happen? It also turns out that this ascorable radical can be formed whenever there is any form of oxidative stress in the body. So if there's some kind of a damage to your phospholipids of your membrane, if there is a problem with your biochemistry intracellularly, if there's inflammation, if there's detoxification going on, this NOS is nitric oxide. It's a type of activates blood vessel dilatation. All of these factors can be oxidative stress, and every one of them initiates the production of the formation of the superoxide molecule, which then is responsible partly because the vitamin C in your system is able to form this ascorable radical. So it's a universal response of the body to oxidative stress. It's almost like when there's oxidative stress here comes the activated form of vitamin C, the ascorbyl radical, which is our redox buffer molecule. It's there to help calm things down and to restore the cellular milieu back to homeostasis. So that's really what the molecule's doing, is helping maintain biochemical homeostasis. So basically what I'm saying here is ascorbate radical formation may be induced by nearly all reactive oxygen species intrinsic to the biological environment. So we're beginning to see just how important this molecule is in terms of maintaining balance. Now, this is I'm going to go to the second part of the presentation of the article that was published, where they showed what happened in the extracellular fluid. It was similar. If you look down here, you do have hydrogen peroxide forming in the extracellular space. But the ascorbyl radical, when it donates its electron here, it is converted to dehydroascorbate. And dehydroascorbate has a molecular shape very similar to glucose. As a matter of fact, the cells in your body will oftentimes mistake the dehydroascorbate for glucose. And if it's a cancer cells are very hungry for sugar. If you doubt that, the PET scan is what a lot of doctors use to diagnose cancer. They'll inject this radioactive tagged glucose into your body, and wherever it starts to glow, that's where the cancer cells have taken up the sugar in very active form. And so here's your glucose over here, and here's your dehydroascorbate. Actually, this is the vitamin C molecule, but they're close enough that the cancer cells will take up the vitamin C. Now, the reason I wanted to show you this is there is now research coming out of Cornell. And this was presented in Kansas City at the KU Med Center last September, and I was there. And this is the Trojan horse theory of how vitamin C helps certain types of cancer. They are specifically researching colon cancer. University of Iowa is really looking a lot into pancreatic cancer. And then the KU Med Center has done published research on ovarian cancer. But they're all kind of looking at this same mechanism, that when the vitamin C radical is converted to dehydroascorbate, which looks like sugar, the cancer cells, which have upregulated receptors called GLUT receptors, will really drink in this form of vitamin C. And the dehydroascorbate will be converted back to regular vitamin C, which can then act as a way of generating more intracellular hydrogen peroxide, which can then kill the cell from the inside out. And this is being very legitimately researched now by a number of institutions, even though we've been using the approach and that Dr. Riordan with the RECNAC team had shown it 25 years ago. Now conventional medicine is kind of getting on board and seeing that this is a plausible mechanism for how vitamin C can help out with cancer patients. Okay, so asking the question, how does the body create this ascorbyl radical, or it's not the ascorbyl radical, the activated oxygen? One of the interesting things is you need iron. But where do you find iron in the body? Obviously, you find it in the hemoglobin. There's iron in the hemoglobin. But it turns out that within the electron transfer chain that I've showed you, most of those cytochrome molecules, cytochrome 1, 2, 3, they are containing heme. That's iron. So they are donating electrons to iron, and the iron is helping with the electron transfer. Okay? So these are the cytochrome 1, 2, this is Q is CoQ10. This is how your CoQ10 is being used in your body. It's being used in the electron transfer chain. And then finally it goes into 4, and then that is reduced. It's the oxygen down here is what accepts the final electron. If you don't have oxygen, you have nothing to accept the electron. There's nothing to pull the electrons through this system. But in the process, this is how the chain works, is through the transfer of electrons down these series of enzymes. So what happened 2.5 billion years ago when we developed the eukaryotes, The eukaryotes are basically these complex, very large, single-celled organisms, which our bodies are composed of. We are eukaryotes, because in the eukaryote cell, it's not only the membrane, but we've got a nucleus. It's got a little membrane around it. We've got all these organelles, ribosomes, and endoplasmic reticulum, and lysosomes. So this made the systems much more complicated and much bigger. I was surprised to find out that a human cell is about 10,000 times the size of a bacterial cell. So it was almost like the little villages became mega cities. And within those cities, a much greater complexity of life developed. And so it evolved on this basis. Life is a chemical system capable of Darwinian evolution. Carl Sagan said that. And the reason this happened is at some time, we don't know when, one of the larger bacteria engulfed one of the smaller bacteria, and that smaller bacteria, instead of being eaten, it enclosed itself in that membrane while keeping the membrane that it already had, and then it evolved into a mitochondria. This is called endosymbiosis. This is how they think the mitochondria at some point developed a symbiotic relationship with another bigger bacteria. And the deal is that the mitochondria could make energy so that the other cell could do other things. It could evolve in other ways. And so this was the beginning of what's called oxidative phosphorylation. And through this evolutionary jump, instead of making 2 molecules of ATP per molecule of glucose, now the cell could make 38 molecules of ATP. The energy production of the mitochondria was so much higher. And so the cell was able to evolve, and cells were able to become specialized, like nervous system cells, heart cells, muscle cells, liver cells, skin cells. These are all eukaryotic cells that are regulated by DNA, but the energy source that drives this evolution comes from your mitochondria. And without the electron transport chain, you could not generate this energy. So again, I'm going to save this for a second, but there's the 38 ATP per glucose molecule. So a big increase in energy production, all because of electronegative power of oxygen. So without oxygen, this would have never happened. So this was the greatest evolutionary step in life on the planet, was the development of the eukaryotic cell's ability to generate all this extra energy through the mitochondria. And it happens because you can think of this oxygen as being kind of like gravity. It's pulling these electrons to it. But and this is based upon what's called redox potentials. I'm not going to get into them, but the high redox potential of oxygen is what fuels electron transport, and this is how we're able to derive the energy from our food, is through electron transfer. So and then there's a number of molecules that these reactions occur because of that. So this is our Irwin Stone, just a little segment of his little quote. On a molecular basis, the whole living process is nothing more than an orderly flow and transfer of electrons. When you start getting into biochemistry and physiology, it's about all redox and the proper use of electrons. And when you're tired, you could say that your electrons are not flowing in an orderly way. You're not able to generate energy. And then that can give rise to chronic inflammation and chronic fatigue and other chronic diseases. So just to make this kind of thinking pretty clear in your mind, you can think of what's happening as a burning candle. So when you put the glass jar over the burning candle, the flame goes out after just a little bit. And why does that happen? No oxygen, that's right. So the oxygen will be used up, and so then the fire goes out. But wait a minute, the flame was still there, and the wax was still there. Wax is hydrocarbons. It's kind of like oil or gas. These are hydrocarbons, and that's what's happening, is they're being burned. But why did the oxygen get used up in the flame? So ponder that, and we'll come back to this answer in just a minute. But these redox potentials is what drives electron transfer. And without electron transfer, there's no life. You've got to have electron transfer, so you've got to have these redox potentials going in order to generate the energy you need to make the molecules that your body needs. And so this particular molecule, which is NAD, was derived from, it's vitamin B3, it has to be at a ratio of 700 to 1. It's an oxidant. It wants to grab electrons and hydrogens from NADH. And without that grabbing power, your mitochondria is not going to work properly. So this is a very simple schematic of what the electron transport chain looks like. And it basically, what I'm saying here is that all life is powered by electronegative electron transport mechanisms based upon these redox potentials. So probably there are all kinds of schematics showing you what the electron transport chain looks like. But just keep in mind that oxygen is the ultimate electron receiver. That was the great leap here on the planet when oxygen became present in the atmosphere. This very reactive molecule allowed for the cells to make this leap in increasing the amount of energy that we could derive from glucose. So let's go back to our analogy. Why did the flame go out? The redox potential of the hydrocarbon bonds could not be activated because the excited electrons, even though they were heated and burning, they had no place to go because the oxygen as the ultimate electron receiver was used up. It was converted into water and carbon dioxide. So it's just the opposite of photosynthesis. Photosynthesis takes water and carbon dioxide and creates sugar. And then when you burn sugar, carbon and hydrogen, you release the energy, but you have to have the oxygen there as the electron receiver, which it receives it and it basically turns it back into water again. So it's so interesting, water and carbon dioxide. So here's what I was mentioning earlier, that atmospheric oxygen is a relatively poor electron receiver. It's not as reactive as we tend to give it credit for. Yes, you need oxygen for something to burn, but you have to have something to burn for the oxygen to really burn. Oxygen itself does not burn. So and this is technical, so just bear with me. Atmospheric oxygen is a biradical with two unpaired parallel spin electrons. And based upon a physical chemistry principle, the Pauli exclusion principle, atmospheric oxygen in the ground state is usually non-reactive to organic molecules. And that's kind of good because otherwise we'd be catching on fire and burning up because we're organic molecules. Organic molecules have paired electrons with opposite spins. This is why biochemical reactions involving electron transfer of only a single electron, that's the only way that it's going to work. And that's why we're going to find out why vitamin C is so important. So in order for atmospheric oxygen to become reactive and useful, it has to be activated up to the superoxide form. So now it's got this extra little electron. And then through a series of reactions, it becomes hydrogen peroxide, the hydroxyl radical, and finally becomes water again. But you can see there's a lot of energy here. This is a very high energy situation. So the activation of oxygen is a crucial step in helping your body utilize the oxygen properly. And so we can do that with vitamin C in an iron environment, or we can do it through the electron chain. And so Dr. Levy, my good friend, says that vitamin C is a good source of energy. We don't think of it as food, but vitamin C does provide electron flow when it reacts with iron. So this is a way of creating the activated oxygen species. And if you keep it going, if you keep infusing high amounts of vitamin C, or if you take your vitamin C orally throughout the day, you can get even higher levels of activated oxygen species, the hydroxyl radical. This is why we find that an IV vitamin C can be more effective in treating an infection than just oral vitamin C alone. So the electron transfer chain itself catalyzes oxygen to the superoxide state so that it can receive the electrons. Otherwise, the chain wouldn't work. Now, what's happened is that most of your chemists think of this superoxide as a reactive oxygen species, that it's a bad guy, and that it's the reason we age. We basically rust as we get older because of these reactive oxygen species. You've heard of free radicals. It's the free radical theory of aging and disease. But what about the activated oxygen species? Is there a place for that in better health and better energy? And I think there is. So this leads into the third part of this lecture. Is everyone still with me? I'm sorry I'm going so fast. But we're now going to talk about how this is what we just finished. Activated oxygen creates the mitochondrial electron flow. And so the theory of how these free radicals create disease is it's under the term electron leak, that there's a leak of electrons from the electron transport chain, and that these are overwhelming the protective intracellular antioxidant enzymes, SOD, catalase, and glutathione. And so the cell's defenses are being flooded, kind of like the way the Florida and Houston, they were overwhelmed with too much water. Well, you can get too much electron leak, and that can cause damage to your mitochondria. This is where vitamin C comes in. Vitamin C can soak up the extra leak, the extra electron leak. It can buffer it. It can help take it somewhere where it's needed. So keep that in mind in terms of understanding how the ascorable radical may be nature's stress adaptation molecule. It's a redox oil, and it helps with the dealing with oxidative stress. So, and this is actually Irwin Stone's comment. It acts substantially like an oil for the machinery of life. And it was discovered by nature billions of years ago. He's found evidence that vitamin C was part of the early process of eukaryotic life here on the planet. So now we go into the third part of this lecture, which is how to repair early onset mitochondrial dysfunction. Early onset mitochondrial dysfunction was a term that was coined by Dr. Frank Schallenberger, who's a friend of ours, and he's spoken at our conference. He deals a lot with oxidation in the form of ozone and ozone therapy, but he was able to show how most of your chronic diseases like cancer, heart disease, autoimmune disease, depression, rheumatoid arthritis, just about anything you can name, obesity, all of these stem from early onset mitochondrial dysfunction. When you have brain fog, when you can't think, if you've got Alzheimer's, if you're tired, if your muscles ache, these can all be indications that your mitochondria aren't working like they should. So the question that I'm asking for this third part of the lecture is, what do we do? How do we repair that? What is the secret to improving mitochondrial energy? Well, the secret is you've got to repair your mitochondria. You've got to make sure they're working smoothly. So this was part of a presentation that I gave up in Toronto earlier this year, talking about a unified theory of chronic illness, because there are, not just me, but there's about 10,000 papers a year being published on mitochondrial dysfunction and how mitochondria may be the secret to why our society as a whole is struggling so much with chronic illness. And so, and we'll be talking more about that, IVC, Nature's Remedy for Chronic Disease, next month when Dr. Levy is here. But I've created a simple little diagram over there. You can see it's got a little mitochondria and an electron transport chain in the middle of it. But if you think about what happens with any illness, and this is more the acute phase of an illness, we're walking along and everything's fine, and we step on a nail or something like that, or we're pounding in a nail and we miss and we hit our thumb. And so we go from that adapted state of health into a damaged state. And you can look at all the different ways that we get damaged. We get damaged by infection. We get damaged by excessive sunburn, or we get damaged by smoke or cigarette smoking, or molecules, things in food that shouldn't be there, or environmental chemicals, carcinogens, all kinds of ways that we get damaged in the world. I truly believe the world now is probably about as toxic as it's been. So we're all trying to deal with that. But at the cellular level, when there's damage, there has to be a signal to the information apparatus, the DNA, and the inflammatory apparatus, and the healing apparatus of the cell. There's got to be a coordinated signal that initiates a repair process. And if we have the necessary resources, if we have the reserves, as Dr. Riordan says, then the body can go ahead and heal. But chronic illness, by definition, are illnesses that are not healing. They're chronic. And you can go to the doctor, and more of the pharmacology of what's done is not so much about healing the condition, is as kind of dealing with the symptoms of the condition so that it becomes a little bit more livable. But what we're really interested in, what does it take to actually do the repair? And that's why I wanted to create this theory. And so you can see that in terms of what throws us into damage is two things: external oxidative stress and internal oxidative stress. And when Dr. Levy's here, he basically says the whole fundamental damaging effect in life occurs through excessive oxidative stress. There's internal and external. That causes the damage. And the repair occurs if we have the reserves, but if we're depleted, we don't repair very well. So the types of damage that we can sustain can be structural damage to the cell membranes, to the DNA, to enzymes. It can also be chemical through those mechanisms. And here's my little contribution. Between signal and repair, there are two ways to look at oxidative stress. One is the reactive oxidative stress, which is where it's actually doing more damage. The repair process can actually do more damage, and this is a lot of what, the body attempting to repair arthritis. Let's take rheumatoid arthritis. That's an autoimmune disease, and that chronic inflammation is actually doing more damage in its effort to try to repair itself. And this is why conventional medicine gives basically steroids and immune blockers to people with autoimmune disease. So we're just trying to calm down the repair mechanism. I'm going to say that the activated oxidative stress, AOS, may be the way that we really need to go. This is what the body is trying to do, but it doesn't seem to have the resources to do it. So what I did in my little chart was draw these complicated diagrams, these triangles, one showing what would be examples of activated oxygen species, and then the red one, that's the green one. The red one over here would be reactive oxidative stress that would be causing more damage than it's doing good. So I won't have time to go into these, but this is what Dr. Levy and I are going to be talking about at our next conference, is that the green upward-facing elements are the factors that can help restore balance to this process of oxidations. So we need to stop looking at oxidation as a foe, but really what's happened is we've lost the balance between oxidation and reduction, or antioxidants and oxidants. There are doctors out there that think antioxidants are the cause of disease. Well, that's why do we eat? Why do we eat foods that are rich in antioxidants? We need antioxidants, and they're actually saying maybe we need more pro-oxidants, but that can be a problem too. So I think the answer is balance, and I'm going to show you how we can measure that in just a minute here. These reactive oxygen species, if you look at these, like the methylation cycle being out of balance, sleep quality being inferior, hormones out of balance, nutritional deficiencies, chronic infection, chronic toxins, chronic inflammation, not being able to burn fat properly, poor oxygen utilization, you're not getting enough exercise, your thyroid's out of whack, you've got insulin resistance, fatty acids are deficient, you don't have enough glutathione, you have chronic stress, and this down at the tip of the spear, NAD+, this has to do with the chemicals within your mitochondria are not strong enough to drive the process forward. They have to have a certain ratio of 700 to 1. So these are examples of the opposite of what we've said for years here at the Riordan Clinic, that there are multiple causes that have to be identified as part of the healing process. And part of what we do here when we do the Real Health Discovery Panel is that we're helping look at the individual to figure out which elements are out of balance. For years, Dr. Riordan said you can't just use a single nutrient to heal people. So I'm saying right now that vitamin C by itself is not the whole answer. It's a very powerful treatment, but you have to look at the whole person and correct on a holistic basis in order to really bring about true healing. So that's our mission, is to help you create real and lasting health by identifying, you have to identify them, and then begin correcting the hidden root causes of your chronic illness. So these root causes come about in very common problems that we see over and over again, and it's a process of reorchestrating your diet, your supplements, your exercise, your sleep, your thoughts, your spirituality. If you need, we're not really spiritual counselors, but we are interested in your spiritual wholeness, that being a part of this. Hormone balancing is very important. Digestive is huge. Eating the right kinds of foods, a clean environment. All of these things can be a part of the process of getting well. And notice that inflammation, adrenal fatigue, and toxins, vitamin C plays a very important role in all of these. Matter of fact, vitamin C, if we stopped and talked about it, it can help out in every one of these. But by itself, it's not the whole answer. Now, my good friend Frank Schellenberger, he came to the same conclusion, but from the opposite angle. We think of vitamin C as the ultimate antioxidant, so to speak. He uses ozone, which is the ultimate pro-oxidant. And the way we got to starting to talk to one another is that he found that if he used vitamin C IV in conjunction with his strategies for ozone, he could get better results. And so we started to see that, hey, we need the redox approach. Redox medicine is using both oxidation and reduction antioxidants as a way of modulating these common denominators of chronic illness. And so if you are not balanced, you're going to end up developing what's called cellular hypoxia, and that's going to lead to mitochondrial decay. So the first thing that Frank did that was such a breakthrough is he said, I've got to measure this in people. And this is Dr. Riordan's mantra as well. We've got to measure before we can, begin to make any kind of an intervention. Otherwise, you're just stabbing in the dark. And so he found a way to analyze how oxygen was being utilized at the level of the cell, at the level of the mitochondria. And he called this bioenergy testing, where he was able to determine an energy quotient for his patients. And this measures oxygen consumption and carbon dioxide production. Oxygen consumption is related to mitochondrial efficiency. Carbon dioxide production determines how well you're metabolizing fat. And so he found that cellular oxygen utilized would create carbon dioxide, which was being produced by the citric acid cycle, which is part of the mitochondria. The greater the amount of oxygen utilized, the greater the amount of CO2 produced, and that there was a one-to-one ratio. This is just the citric acid cycle. So he was able then, looking oxygen going in, carbon dioxide coming out, and create this number called the energy quotient. So without going into this graph in a lot of detail, basically what it shows is that if you're using oxygen well, if your mitochondria are functioning efficiently, what you'll see is a 1-to-1 or 100% utilization of oxygen being used by carbon dioxide. If you, however, got into anaerobic glycolysis, if you're running down the road and all of a sudden you get a side ache and you're acidy feeling in your mouth, what's happened, you've gone into glycolysis and lactic acid is being formed, and that is a much less efficient way of generating energy. And so what happens in the readouts when he does the oxygen carbon dioxide measurements, he sees more carbon dioxide being produced. And from that, he can give that a number. So when you have more smoke, more carbon dioxide, that's poor oxygen utilization, and your body is creating a lot more reactive oxygen species. He was also able to use this same calculation to determine what percent of your metabolism was burning carbs and what percent was burning fat. And so this is all laid out here. And if you get a chance to revisit this video, you can look at this and you can see how he was able to calculate fatty acid metabolism versus glucose metabolism. And you can see it right there that fatty acid metabolism is much more efficient than glucose metabolism. So the glucose metabolism takes place in glycolysis. The fat metabolism takes place in the Krebs cycle, in the mitochondria part. So he was able to show what percent you were fat burner versus carb burner. And if you were more of a fat burner, you were more likely to have a great oxygen utilization. If you were more of a carb burner, which I think happens to a lot of people as they get sicker and sicker and tireder, they start moving more towards carbs and sugar, craving sugar, like cancer patients crave sugar. What's happening is you're pumping more sugar into the inefficient way of generating energy. And the efficient way, which is your mitochondria, are being used less and less, and they begin to decay. So you start getting into problems with that. And this is where vitamin C can help. Won't go into that. It helps your adrenals. I won't go into that right now. But in general, when you're using oxygen poorly, you're going to be expressing more illness. When you're using oxygen optimally, you're going to make better use of your nutrients. Your intracellular antioxidant enzymes are formed. You make this NAD, which is so important, so that you can generate ATP, which is your energy currency. We generate our weight in ATP every day, but it's recycled in order to keep us going. And so these are all about healthy lifestyle choices. Whereas you can, and this is this is where you're creating an intracellular milieu that is balanced. So I won't go into more of that. So this, I'm 1 minute away from finishing here. So catch my breath here. So really, this is, if you start to see that we have an amazing repair system in our body, but it depends upon energy, and that depends upon proper oxygen utilization by the mitochondria, which needs to be regulated properly, and one of the key regulators is vitamin C, but that's not the only regulator. All of these factors over here in the green, these are very important regulators to help us keep our oxygen utilization under control so that we don't have free radical damage in the cells. So so our next lecture is going to go into these lifestyle choices and why they are so important as part of nature's remedy for chronic disease. Dr. Levy is an expert in vitamin C, so we'll be emphasizing heavily the advantages of IV vitamin C, but we'll be talking about how IV vitamin C by itself is not the whole story. You have to really balance your overall health and lifestyle in order to get your mitochondria up and running in an optimal way. So at this point, thank you very much. I'm sorry I had to go so fast. Let's take some questions. Is the microphone still here? I think Dr. Anna, do you have the microphone? Here we go. And don't feel intimidated by all the verbiage. I apologize for having to introduce. Probably there are slides and there are graphs that you've never ever seen before in your life, and you're thinking, what in the world is he talking about? But again, this is where we are getting down into the subcellular level. How do our cells generate energy, and what's happening that's causing us to feel low energy, and what can we do about it to begin to restore ourselves to better health? Let me give you a couple things. Aerobic exercise is a very good way to begin utilizing oxygen better. And just about everyone agrees with it. Now, the problem I have with it is a lot of people exercise too much, and they cause more oxygenation through their body and oxidative stress, and they end up getting hungrier, and they end up making poorer food choices. They don't lose weight, so just exercise alone isn't going to do it. But exercise done appropriately is a very powerful healing tool. Getting the right kinds of foods, very powerful healing tool for your mitochondria. You know, having some days where you don't have any sugar at all, you're basically coaxing your mitochondria to shift back into fat metabolism. This is one of the ideas that's coming out now, is that of intermittent fasting. And our ancestors probably intermittent fasted all the time because they were hunter-gatherers. The meat was not always there. They couldn't find the food to gather. And so they might go a day without eating or longer. They didn't have a refrigerator to go to in the morning when they got out of bed. And so there might be a 24-hour period where they didn't eat at all. What happened then is that they had to shift into fat burning. And so having that mitochondrial flexibility, regaining that ability to not eat and not feel totally wiped out, because some people, if they miss a meal, they feel like they're starving. if you can shift back from glucose to fat to glucose, you're going to feel a lot better, have a lot more stamina, and it's going to be a lot more easier to gain weight. Because when you're shifted into glucose metabolism all the time, you tend to develop insulin resistance. You tend to put on weight. You tend to feel sluggish. You don't sleep as well. And so you start to see a domino effect to where you're kind of trending down towards poor health. And our goal here is to trend towards better mitochondrial functioning, better oxygen utilization, better food choices, better sleep. All these things tend to kind of domino you back in the direction of health. Health, in my mind, is addictive. If you start feeling better and you know and you've made a change in your life that helps you feel better, you'll tend to stick with it. But you have to have an idea of why you're doing it, what you're doing, because usually these things don't happen the first time you try it. You have to kind of like retrain your body to utilize its metabolism in a better way. Yes? Here comes the microphone, yeah.
When you talk about no sugar days, do you mean, I know you mean the sucrose, but do you also mean the fruit?
Okay, so the question, I'm not sure if it went through on the tape, but when we say low or no sugar days, does that include fructose? I would say low glycemic fruit in season and ripe is probably a pretty good choice. The problem we have in America is that we have what apparently ripe fruit that's been flowing in. It looks great, no spots or anything. And so the idea is to, a lot of people say eat more fruits and vegetables. I would say eat more vegetables and some fruits. So we're not demonizing fruit, but fruit and the fructose that's in it is more likely to be stored in the liver and cause some insulin resistance if you're getting too much. So I don't think we need to go overboard on fruits. And I would always choose the low glycemic versions. And there is a nice video online that Dr. Ann did about is sugar bad for you? And it's not if you get it in the right way, the right timing, the best quality fruits.
Thank you.
Yeah.
I came across a reference that forms of light can be unhealthy for your mitochondrial activity, and in particular that LED light can damage mitochondrial activity. How does that work?
Well, I'm not an expert in that. I know Dr. Riordan had a lot of interest in light and light therapy. The best book reference I can point you to is called Headstrong by Dave Asprey, and he's got a whole chapter devoted to getting the right kind of light and using maybe even special glasses in order to block the blue light. And so all I would know is that I don't feel good in a fluorescent lit room. I know that. And the LED light doesn't seem as good as natural lighting. So, but there's evidence now that it does affect mitochondrial functioning. And it's part of a comprehensive plan to reinvigorate your mitochondria. But it's a little bit more than what I'm expert in. So, but it's a good thing to think about.
How do you measure for this dysfunction?
Okay, so the question is, how do we measure for mitochondrial dysfunction? And it turns out that this EQ testing that I showed you is a special set of equipment that's very easy to do. It's a software program. You basically put on a mask, and it's measuring the amount of oxygen you're consuming and the carbon dioxide you're producing. He measures it at rest and at exercise. And then from that, he can calculate your maximal oxygen utilization and find out if you're below the predicted level for your age. If you are, he says that's very good evidence for early onset mitochondrial dysfunction. You cannot correct that by just exercising more. You have to start getting at these root causes and correcting those. Because otherwise, and he did a test on 50 young men between the ages of 20 and 40, young men, and found that almost half of them, not quite half, 46% already had early onset mitochondrial dysfunction. So this is different than disease, but it's the precursor to disease that will get you stuck in a low energy state. So one of these days I'm going to get up there and get the training on Schallenberger has a course on how to implement this. We just don't have it here at the Riordan Clinic.
One thing that
Mercola says that if you want to kind of measure your aerobic ability, take your age and subtract it from 180. And that's your target pulse rate when you exercise. So if you go out for a jog and your pulse rate gets higher, then you need to slow your pace down. And eventually what will happen as your body learns to adapt and you utilize oxygen better, you'll be able to go harder and faster and still keep your pulse rate low. So aim for, 180 minus your age. So if you're 50 years old, you don't want your heart rate getting any higher than about 130 when you exercise.
Keep in mind, though, you cannot exercise this early onset mitochondrial dysfunction away. Exercise is very important. It's part of the healing process, but there are other things, your diet and other things like this that need to be hormonal balance. Other things need to be taken into consideration to actually reverse this EOMD, which is the initials for that.
We have a question through our YouTube stream, and the question is, when is it appropriate to give glutathione to a cancer patient? Some say this competes with the oxidation, but you are saying we need a balance.
Well, our bodies are making glutathione all the time, and in general, we need glutathione. The big question is, based upon that little bit of research that I showed you about when you give high-dose IV vitamin C, it generates extracellular hydrogen peroxide, and then that hydrogen peroxide then can be converted to the hydroxyl radical, and we think that's what's killing cancer cells. Well, we don't want to neutralize the hydroxyl radical too much with excess glutathione. So this is still a question that's being researched, but generally speaking, most people who follow the Riordan IVC protocol, we recommend not giving the glutathione in a cancer patient, though by the same token, we do correct methylation cycles so that they can make their own glutathione more appropriately within their cells. Okay.
I was in not very long ago when I had the, is it the UVB light? Okay. I did notice that, and it hadn't happened to me in the past, but my blood actually just looked fake. It was kind of purpley and just way different than normal. Does that mean I'm not oxygenating my blood? I mean, would it help to keep going with get the C going? Or does it just mean I'm sluggish?
Good question. I mean, I think there are people that have looked at blood as a way of kind of diagnosing how well you're oxygenating. There's no question that when we take the blood out for the UVB light test therapy and put it in the bag, if we add ozone or if we add hydrogen peroxide, which converts into oxygen, the blood will become cherry red. It will oxygenate much better. And so chronic illness may be under oxygenation of the blood. It's just a big question as, hyperbaric oxygen is being looked at as a plausible therapy for cancer using IVC, hyperbaric oxygen, maybe the ketogenic diet or versions of the ketogenic diet. All that is out there as ideas, hypotheses that might be useful in the cancer patient. We don't know, but I know oxygen can be toxic too. So you just can't just go live in a hyperbaric chamber at 100% oxygen. That wouldn't be good. So there's something about cycling. There's a cycling factor that has to be taken into consideration. And I think this is what the next 10 years is going to tell, hopefully sooner than 10 years, but within the next few years, we're going to find out a little bit more how to use oxygen appropriately with vitamin C and diet in order to reverse cancer. All right, well, thank you everyone for coming today and for sitting through that highly technical lecture. I hope it was helpful to you.
You guys please fill out the surveys, and if there are any lectures you guys want in the future, just write those at the bottom in the comments.